All right, welcome back. Here is video number 2. This is going to be the first one where we've actually got a short series within the same concept. So we're talking about cells, right? The basic building blocks of life, okay? All living organisms have them. Some living organisms are only one of them. And cells have a lot of things in common. And so in this series, we're going to talk about the different organelles. Now we're focusing on animal cells. So a couple of things, right? We're only talking about eukaryotes. So no bacteria. We're only talking about animals. So no plants, no cell walls, none of that stuff. Okay. Humans are animals. We're going to be talking about animal cells. So if you are looking forward to some of that other stuff, you may need to go back, maybe do some Amoeba Sisters or something like that, okay? Some crash course biology. We're talking just about animal cells here, okay? And so it all starts with cell theory, right? And so like I said, cells are the building blocks of all living organisms. One of the sort of, I guess, requirements of being considered alive is you are a cell or you are made up of cells, okay? Cells come from the division of pre-existing cells, which gets into a lot of really interesting conversations about where did those first cells come from. A lot of thinking is sort of going into the formation of self-replicating molecules that just started to get more complicated. There's a lot of research if that's the sort of thing that you're interested in. A lot of good stuff out there about the origins of life and things like that. Cells are the smallest units that can perform all vital physiological functions. Okay, so we're talking about moving, taking in food, avoiding things that we don't like, eliminating waste, all of that stuff that we do. We breathe, we eat, we get rid of waste, we try to replicate. All of that stuff happens at the cellular level. And then as you add more cells, you get specialization and complication and stuff like that. And each cell maintains its own internal homeostasis at the cellular level. Now, if you remember way back at the very beginning, we talked about homeostasis, right? Maintaining a favorable internal environment, keep the outside in, keep the inside in, keep the outside out, right? And keeping things favorable, maintaining that liquid environment, that internal sea sort of thing. So when we're talking about cells, we're talking about cytology. Here we go again. We got some words that we're breaking down, right? Cyto meaning cell, ology meaning studying. So cytology is the study of cells. See, I just told you that. I'm so smart. When we're talking about cytology, we're talking about different types of cells, what they do, and stuff like that, right? And so there's 2 different types of cells, right? Sex cells, also known as germ cells or reproductive cells, and those are the ones that are responsible for creating new organisms, right? In male, we've got sperm. In female, we've got oocytes, also known as eggs. And we're going to talk a lot about those way at the end of the course. Talking like May-ish, okay? Last thing that we talked about is basically where do babies come from? So if you've ever wanted to know, I got you, all right? Then the other type of cell are somatic cells. That's basically every other kind of cell, okay? All the body cells except for the sex cells, okay? So now we're getting into the organelles. Now I posted in Echo, if you wanted to use it, a helpful note-taking guide that lists all of the organelles that we are going to be responsible for. I'm going to go into way more detail than you can fit in those little boxes. However, it's a lot of really good information. And a lot of this stuff we'll hit on as we go. For example, the plasma membrane is going to come up again when we do transport. All right, so our cell membrane, plasma membrane, keeps the extracellular fluid, also known as the interstitial fluid. Now, I'm going to apologize right now because from here on out, I'm going to be stumbling over words and stuff like that, okay? So please forgive me if it takes me a couple of chances to try to say some of these words. Plus, I'm doing it all in one take. This is raw, one take action that you're seeing here. So what is happening now is happening now, except if you're watching this later, because then it was then. Anyway, it doesn't matter. So the interstitial fluid is the water around the cell, basically, right? So it's kind of similar to what the water inside of the cell is. It's just on the outside. And so the plasma membrane is going to separate the cytoplasm, that's the fluid inside the cell, from the extracellular fluid. Because if that plasma membrane's not there, now that's kind of one of the ways antibiotics work. If you break that plasma membrane, everything is just sort of floating around. And so they can't really function together as a unit, and so the cell is dead. And so here's our cell. The plasma membrane's in the pink. The non-membranous organelles, and I'll talk about that in a little bit, is orange. And then the ones that have a membrane around them, so that they're kind of their own little pocket, are in the purple or the blue, that sort of thing. And so we're focusing primarily on the plasma membrane, right? So different functions, right? Because you need to know that. Protection, isolation, sensing the environment around them. That's what these little things do. There's also signal proteins, support, and regulating what comes in and what comes out. Okay. Let's see, which I think I just mentioned here. So I'll go through these really quickly because I just mentioned them. I apologize. So it acts as a barrier. What else? Regulation of exchange, stuff coming in, waste going out, sensing the environment and moving away from bad stimulus or based on what the interstitial fluid is like, it's going to change the actual inner workings of the cell to take in or get rid of stuff based on those external signals. Structural supports, what sort of gives the cell its shape. The cell membrane is also what allows the different cells to stick together to form tissues and then organs and stuff like that. So here's our plasma membrane, okay? And so we've got the main component. Now remember from the macromolecules video, I said the word phospholipid, and I said it was going to come up in plasma membranes. Well, boom, here we are. Talking about organelles, talking about plasma membrane. So our phospholipids form a bilayer. So a bilayer, also means two, okay? Bilateral is two sides. Bicycle, okay, two wheels, two cycles, right? The phospholipids are really important types of lipids, okay? They've got two parts. They've got the hydrophilic heads. Okay, hydro. You remember this back from when we talked about dehydration synthesis and stuff like that. Hydro meaning water, right? Philic means you like something, you love something, okay? Hydrophilic, water loving, okay? Maybe write that down because it helps. If you can get those context clues, you can get those vocabulary clues, it helps you with identifying what's going on. So hydrophilic, water loving. And then you've got tails, right underneath. That's what these things are right here. Hydrophobic, okay? If you are arachnophobic. Does anybody know what that is? Give you a second. If you're arachnophobic, you are scared, phobia, of spiders, arachnid. Agoraphobia, you're scared of the outside. Triskaidekaphobia or something like that is you're scared of the number 13. So hydrophobia is your, I guess, keeping with the same content, you are water fearing, right? You want to stay away from water. Oil is hydrophobic. They don't mix together. Because then that gets into polar and non-polar and stuff like that. Water's polar, oil is non-polar. Chemistry stuff. If you want to go into greater detail, we kind of talk about a little bit more when we do blood and sort of what gets exchanged and stuff like that. And so it provides a barrier to water-soluble compounds and ions because we don't want those just randomly flowing in and out. And we're going to look at why when we do cellular transport and osmosis, okay? That means certain things can dissolve across the membrane, okay? Also within, excuse me, also within the membrane itself, we've got integral proteins. So integral means it's a part of it. It's embedded within it in this particular case. Okay, if something is integral, it's vitally important. Okay, so integral proteins are proteins that are within the membrane, and you've got a lot of different types of proteins. You've also got peripheral proteins. Let me see if I've got an example of that one. So here's one right here. Okay, so a peripheral protein is either stuck to the inside or the outside of the membrane, but it doesn't cut all the way through it like those integral proteins do. Anchoring proteins are acting as stabilizers. Let's see, I don't have an example. So anchoring acts as a stabilizer. Recognition proteins, these are your glycolipids and your glycoproteins down here. Those are... hold on, let me back up. Your glycoproteins are something a little bit different. Okay. Your recognition proteins, I'm going to call those flags a lot, especially once we get to the immune system and the endocrine system. We're going to talk about those identifiers, those signal flags. Those are the ones that basically identify your cell as a U cell and not a something else cell. We got receptor proteins. Those are the ones that are sort of waiting for a signal from the extracellular matrix, the interstitial fluid, whatever you want to call it. And those signals, those chemical signals, are called ligands. Okay, and so that's any chemical signal. Okay, hormones. When we talk about the endocrine system, hormones are ligands. Okay, and then we got carrier proteins. Carrier proteins are specific proteins that are responsible for bringing stuff across the membrane, either in or out. You've got different directions and stuff like that. And they're specific solutes. So like when we do nervous system, we're going to talk about sodium channels and potassium channels, okay? And there's other channel proteins as well, but they basically allow stuff to travel across the membrane that normally wouldn't be able to. What else do we got? Some of it will actually regulate water that goes through, and then specific ones are called gated. So you've got open channels that just allow stuff to go through. That's this one right here. See how there's nothing that's stopping stuff from going through it. Then you've got gated channels, so those are usually there's some sort of mechanism that's going to keep that closed until it's needed to, and then it opens and stuff like that. And we'll talk about those a lot when we do action potentials and nerve impulses and stuff in nervous system. A couple other things then just to point out real quick. We've got specific carbohydrates that are outside of it that act as signaling mechanisms, a sort of coating mechanism. That's the glycocalyx, is a sort of like sugar coat, basically. That's where you have the anchoring part coming in. They act as receptors, recognition, stuff like that, the signal flags and what have you. Okay, so within the cell membrane, you've got the cytoplasm, and that's basically all of the stuff that is inside the cell but outside of the nucleus. Okay, we've got the cytosol, which is the intracellular within, so intra is within yourself. Okay, so within the cell, intracellular fluid. And then you've got the organelles as well. And so these are all the materials. So those ions and stuff that we've brought across, or the waste that we have to get rid of, is just going to be dissolved in the cytosol. We've also got proteins and amino acids and stuff that are sort of floating around that we'll grab those as we need them. And then Not a lot of carbs, lipids, amino acids, and sodiums. And then the organelles, which are the specific structures floating around within that stuff, that cytoplasm, cytosol. Okay. Moving on past that, if you look at the picture, we've got two different types of organelles, I guess you can think about. You've got non-membranous organelles. So they don't have a membrane, basically. There's not like their own little sac or whatever. They're just floating around in direct contact with the cytosol. Your examples would be your cytoskeleton, which we'll talk about in a little bit, ribosomes, same thing, centrioles, proteasomes, microvilli, cilia, and flagella. Cilia and flagella are used for locomotion. Cilia can be also used for sensation. Microvilli increase surface area, and there's used for sensation and absorption and stuff like that. And then membranous organelles are all the other ones, so each one of them have their own plasma membrane. Remember, one of the ways that we are eukaryotes is we have membranes that are separate from the surrounding fluid, that cytosol. Bacteria, they don't have that. Literally everything is just free floating, right? Your mitochondria, lysosomes, peroxisomes, which are really similar, your endoplasmic reticulum, both smooth and rough, and the Golgi apparatus are all membranous organelles. Now, we also talk about the nucleus as an organelle. It's kind of separate, though, because it's got its own nuclear envelope separating itself from the rest of the cell. So in a lot of ways, it's also its own, more of like a membrane-bound region than an organelle. But it also functions as an organelle, so it's kind of weird. What else? Okay, so inclusions can just be masses of insoluble stuff in the cell, like lipid droplets and stuff like that. The cytoskeleton is one of those things that basically provides the framework, the shape of the cell, and basically kind of acts like the skeleton you have in your own body. We've got microfilaments, intermediate filaments, and microtubules. 3 different types. It has to do with how they're structured and stuff like that. So looking at this then real quick, we've got microfilaments right here. All these little reddish, ropey looking things. They provide mechanical strength, so they actually act like ropes. They interact with other proteins within the cytosol, and they also interact with the filaments in myosin, which we'll talk about later as far as muscle contraction is concerned. Then we have the intermediate filaments, which are like thicker strands right there. Those are strength and shape, stabilized position of organelles, and then the position of the cell. Think of it like the connective tissue of the cell, okay? We'll talk about that's the matrix that sort of holds everything together, okay? Holds everything in place. So it's easier to coordinate stuff and get things done quickly. And then the microtubules, which are the largest one, those are like little tubes or hoses, basically. A lot bigger, a lot stronger. Provide strength, structure, and support. They can also change the cell shape. When we get into cell division, it's the microtubules that are going to be the ones that are actually exerting that pressure on the cell to cause it to separate. And then you also have your chromosomes during the cell cycle will separate by way of microtubules, basically ratcheting the parts of the chromosome away from each other. We've got our microvilli. They increase the surface area of the cell, and they attach to the different parts of the cytoskeleton. We have the centrioles, which are kind of like your ratchets, your pulleys, or whatever you want to call it, the winch. I'll just keep coming up with stuff until one sticks. And so these are the ones that will actually pull the cell apart, basically. And they move to opposite ends, and we'll talk about that later in a couple of videos. Some other ones that are just free-floating. You got the ribosomes. Those are the ones that synthesize proteins. You've got a large unit and a small unit. They're made up of RNA. You've got ones that float freely, and then you have ones that are also attached to the ER. Both of those manufacture proteins. It depends on where you're going to send them. Free ribosomes will put the proteins directly into that fluid. Fixed ribosomes on the ER will actually build them within the endoplasmic reticulum, where they are finalized and packaged and then sent to the Golgi apparatus, and I'll talk about that later. So here's an example of what those look like. Here's your ribosomes with your two subunits. Here's the free ones. Here's the fixed ones. Same ribosomes. Both have subunits. It just has to do with if they're free floating or not. Okay. Like I said in another video, we're not really going to talk a lot about transcription, translation, and DNA replication, so you're not going to have to really know about the steps. This is about as far as we're going to get into that particular setup. And I think I mentioned that later. I don't remember. I've slept since then. Something else, we got proteasomes. Those are organelles that have enzymes. They're basically like sacks of enzymes, basically. And they will actually recycle damaged proteins. So it's proteins recycling themselves or each other. I don't know. It's kind of like molecular cannibalism, I guess. Membrane-bound organelles. We have 5 of them. We've got the ER, the Golgi, lysosomes, peroxisomes, and the mitochondria, which we will talk about all 5 of those right now. First off, here is our endoplasmic reticulum. It has storage chambers known as cisternae. That's these things right here. So here's those folds, right? And this is where the bulk of your protein synthesis takes place, as well as carbohydrate and lipid manufacture. So let's say, for example, in one of the stages of the cell cycle, the cell is getting ready to divide. You're going to make the proteins, the carbohydrates, and the lipids required for cell division here. Okay. You're going to store these materials until you need them, transport them into other parts of the ER, and detoxifying drugs and toxins. Okay. Gotta let that settle down in 3, 2, 1. So we can take the endoplasmic reticulum and we can split it into 2 different types, basically. It has to do with what is attached to it. So we have the smooth ER. It's smooth because there aren't any ribosomes attached to it. And it's primarily synthesis for phospholipids and cholesterols for the membranes, steroids, glycerides, which are the part of the triglycerides that we talked about in lipids, and that's fat storage in the liver primarily. And glycogen, which is sugar storage, which also ends up going to the liver. So a lot of synthesis and storage, preparation for transport of the other biomolecules. The rough endoplasmic reticulum is called that because it's covered in ribosomes, so it looks bumpy, right? And if you actually look at micrographs, which we'll look at here in just a second, it looks rough. And the rough endoplasmic reticulum is primarily responsible for, anybody want to take a guess? Ribosomes. Ribosomes are responsible for making proteins. And so protein and glycoprotein synthesis primarily happens here for the cell. Proteins are also finalized and folded into those different structures, the secondary and tertiary structure. And then they are put into these little packets called vesicles that are sent to the Golgi apparatus. Here's that image that I was talking about. And so if you look at the dark spot in the middle right here, it looks rough. And there's other parts next to it out here where it looks kind of smooth. And so that's why we started calling it the rough endoplasmic reticulum and the smooth endoplasmic reticulum. Here is the Golgi apparatus, also known as the Golgi complex, also known as the Golgi body. I use them interchangeably, so either one of them is correct. If you want to pick one so you know what you're talking about, to make it easier for you, that's fine. Just pick one of the three. This is your like transport hub, basically. So if UPS or Walmart has a distribution center, that's what the Golgi apparatus is. So you're modifying and packaging secretions like hormones and enzymes and stuff like that. Modification of protein, so you're going to add or take away carbohydrates if you need to. Plasma membrane sort of maintenance if it needs to be replenished or something like that, rebuilt. And then the packaging of special vesicles called lysosomes, or packaging the enzymes that are used in lysosomes, which we'll talk about here in just a second. And so lots of folds where storage and stuff takes place, and then basically bits of the Golgi apparatus will fold off and pinch off basically and create these vesicles at this point. Here is a cross-sectional view so you can sort of see those folds. Structurally, it's similar to the endoplasmic reticulum because they there's a little bit of overlap in overall function, basically. Okay, so from the Golgi apparatus, then we have those lysosomes that I had mentioned. They're small vesicles, so if you look at the diagram in the notes page that I had mentioned earlier, if you're using it, the peroxisomes and the lysosomes structurally are basically the same. They look like little bubbles, basically. It just depends on what is inside them, okay? And so lysosomes are vesicles that are filled with enzymes, okay? And so they are primarily responsible for removing damaged organelles and pathogens. So they're kind of like recycling machines, basically. So basically, like super duper quickly, what ends up happening is you've got primary and secondary lysosomes. Primaries have inactive enzymes, so they're ready to go, but they're not going to be used yet. When they fuse with a damaged organelle, they become secondary lysosomes. That makes them active, okay? And then in which case they break down the bacterium or the virus or the damaged organelle and recycle it and chop it up into its pieces and separate it into the different parts that you need. And either A, it releases those materials back into the cell so they can be reused, or if it's waste products, like a bacterium, for example, or something like that, then it takes those and it forces them out of the cell. That's exocytosis, and we'll talk about that in another video. They also will fuse with stuff that's brought in from the outside environment that needs to be broken down, like the lipids and stuff like that. Okay, those don't just dissolve into the cytosol. They have to be worked on, broken down and stuff like that. And so then either they get reabsorbed or the waste gets ejected, like down here at the bottom. That's that part I was talking about. You can also have autolysis. So basically what happens is the vesicle, auto is like within yourself or your own person. Lysis is to break. And so basically this is the self-destruction of the damaged cells. Or of inactive cells. If they are infected with something, if they're damaged, they need to be destroyed. Basically what happens is these lysosomes pop, releasing those enzymes into the cell, and they basically end up sort of chewing up the cell and breaking it down, and then those bits are reabsorbed by the surrounding cells and stuff like that and used to make more cells. This is that programmed cell death, apoptosis that you may remember hearing about from your biology class. It's triggers by these lysosomes. We also have another vesicle called a peroxisome. A peroxisome is another enzyme-containing vesicle. However, it breaks down organic compounds like fats and neutralizing toxic compounds that come into the cell. It basically, what happens is it releases hydrogen peroxide, which breaks down these substances, and then as the hydrogen peroxide breaks down, you get water and oxygen, which the cell uses. So these are basically like cleaning agents. Think of it like your hydrogen peroxide that you're putting on a cut, or a Lysol, which maybe that's not a very good one because the name's not. Okay, we'll come back to that one. But these are your cleaning agents, right? We use that antimicrobial spray on the desks, right? And so your peroxisomes are kind of doing that within your cells, which is kind of neat. A couple more. We got the mitochondria, right? And everybody knows the mitochondria is the powerhouse of the cell. Mitochondria has two membranes, and then that gets into the the theory, y'all gonna have to help me out with this one. The theory that's endosymbiosis, that explains where chloroplasts and mitochondria came from, and the mitochondria may have been a bacterial organism that was surrounded by a larger bacterial organism, and then it just sort of liked it there and it never left. And it's more complicated than that, but that's fine. Basically, you've got a smooth outer one. Inner one, it's got a whole bunch of folds and stuff like that. Those are called cristae. And this is where glucose is, the breakdown is finalized, and it gets turned into ATP. And that's really about all we're going to talk about cellular respiration is concerned. We're not going to go into all of that process like I'm sure y'all love doing when y'all did biology. So you just need to know that the mitochondria is responsible for taking glucose and turning it into ATP. That's what cells actually use, not the actual sugar itself, because it's just too big. And then the last organelle that we kind of need to talk about is the nucleus. It's the largest organelle, and that's the control center, basically. So it has its own membrane called the nuclear envelope, and it's a double membrane around the nucleus. You definitely don't want stuff coming into the nucleus, and you want to control the stuff that's coming out. Between those two membranes is the perinuclear space, and then there's holes in these membranes called nuclear pores, and that's what allows stuff to go primarily out. If you have to make new DNA, the building blocks, those nucleotides will come in from that way also. Within the nucleus is a structure called the nucleolus, which is a kind of like a dark spot. It's the most conspicuous spot in the nucleus, and that's where the ribosomal RNA for protein synthesis is manufactured, and then it leaves the cell. And the DNA, if you can kind of see the sort of squiggles right there if you zoom in, that's the DNA is sort of free-floating all around. And we'll talk about what happens to it and why sometimes it's visible and sometimes it's not when we do the cell cycle. Well, geez, I think as far as the organelles that we're talking about on that list there, that should be all of them. Biggest things that you need to know going into all this stuff. You need to know approximately what do they look like, where would you find them, primary function. I think I said location, where you would find them. Yeah, I did say that. Yeah, so just make sure that you know the organelles that are listed, function, what they look like, where they are, and then you should be good to go. Hopefully a lot of that stuff is review from your freshman year, although I know some of us have slept since then, but hopefully a lot of that stuff came back up to you. If you have any questions about organelles or the notes or anything like that, just let me know. Send me a remind message, send me an email, see me in class, zoom in, that sort of thing. Otherwise, I will see y'all, I guess, when the next video in the series drops. So I'll see y'all then.